Hi everyone, this is your instructor Christina Howard, and today we're going to talk about the lymphatic and digestive systems. So just give me a second while I disconnect my computer and set it in tablet mode, and we will get started. So this presentation is only going to go up to slide 38 because that is where we ended up making it to in lecture before the test, as I mentioned to all of you on Slack. So really we're just going to cover lymphatic and digestive stuff here. So let's get started with that. Off to a great start, already using my stupid pen that I don't want to use. Let's switch to laser pointer. There we go. Okay, so with the lymphatic and immune system, you can really divide this into two different sections. The lymphatic organs and system are basically a way to take the blood and what's in it and filter it, and that helps to increase the likelihood of pathogen detection. So that's really what the lymphatic system is all about. The immune system are populations of cells that basically protect your body. So these are white blood cells of a variety of kinds. We've talked about some of them in the context of the circulatory system, but here in the lymphatic system and the immune system section, we're going to get some more detail about those particular cells. So what they do and where they live and stuff like that. So where the lymph comes from is the capillary bed. So if we zoom in down here, we can see that this is a continuous capillary bed. So that's a concept from the previous section, meaning that it's not fenestrated or a portal system or anything fancy like that. But you can also see that these lymphatic capillaries are sort of intermingled with the blood capillaries. Excuse me. I just saw my dog yawn and it made me yawn. So the blood capillaries reabsorb around 85% of the fluid that leaves. So at the arterial end of a capillary, hydrostatic pressure, which is the pressure of blood pushing on the walls of the capillary, causes fluid to leak out. Kind of like if you poked holes in a hose, right? So increased pressure in the hose, water comes out the holes, you get the idea. Since water is leaving but large plasma proteins and such are staying inside, over towards the venous end of the capillary over here, we have the phenomenon of the colloid osmotic pressure of the remaining fluid in the capillary attracting water back from the interstitial fluid. So that accounts for about 85% of the stuff that left over on the arterial side, which leaves another 15%. And that stuff enters the lymphatic system. So that accounts for between 2 and 4 liters per day. And it's very similar to plasma, but with not nearly as much protein. And that's because, of course, large proteins are too big to squish through the capillaries and they end up just staying inside. That's that colloid osmotic pressure driver. So with regard to immune defense, so these are going to be the cells that actually perform the defense functions. You get defenses against pathogens. So a pathogen, by the way, doesn't have to be quote-unquote a germ. So we think of pathogens as being germs, but they can be non-living problems as well. So infectious organisms is just one kind of disease-producing pathogen. There are others. So toxic chemicals. And I know that everybody's going to think like, oh, like the kind that come from factories and such. Well, yeah, sure. But also like plant irritants. So urushiol is a plant irritant that's found in poison oak and poison ivy, and that definitely causes an immune response. If you've ever had poison oak or ivy, you know very well how painful and obnoxious that feeling is. That's not a living organism at all. It comes from one, but it isn't itself a living organism. Nevertheless, we consider it to be a pathogen. Radiation is also considered a pathogen in this regard. So really just stuff that's going to do you harm. So within immune defense and the population of cells that carry out immune defense, you've got 2 populations of cells overall, and these are responsible for innate and adaptive immunity, respectively. Now, there are some cells that perform both functions, and we'll not talk much about those in this class. It's more for an immunology class, but we will identify some of them. So first defense includes just physical barriers to pathogen exposure. So these are things like having skin and mucous membranes as well. So basically just keeping stuff on the outside and keeping stuff on the inside. The second defense system includes actual cells, but these are cells which are considered to be generalists. So they're not directed against specific antigens as the adaptive immune system is, but rather they're just sort of all-purpose destroyers of pathogens. So macrophages is a really good example. They are avidly phagocytic, meaning that they are big eaters, and they don't really care what they eat as long as it's foreign and has cell surface markers that make it foreign looking, or it's cell debris inside of your own cells and tissues. So they don't discriminate. There are also antimicrobial proteins, so key idea here, non-living. And those are going to basically stick to and disable pathogens, as well as things like immune surveillance, inflammation, and fever, which is called pyrexia. So pyrexia is coordinated by the hypothalamus. Adaptive or specific immunity is called the third defense, and this is where immune memory comes in. So, for example, the reason that vaccines work is because of adaptive or specific immunity. So the way that this kind of immunity works is basically that your body has encountered some manner of pathogen and is able to remember it so that upon second exposure, a response can be mounted much more quickly, such that although the pathogen is technically inside of your body, it never has a chance to make you sick because your immune system knocks it out so rapidly. So that's the major difference. Within adaptive immunity, you've got humoral. So anytime you see that word humoral, kind of think about blood, and specifically blood plasma. The reason that antibodies are part of humoral immunity is because antibodies, also called immunoglobulins, float around in your plasma. Cellular immunity is cell-on-cell crime. So we have really cool sets of cells called killer cells, or natural killer cells. And these cells basically rub up against a foreign cell and destroy it via physical attack. So here's another way to say that stuff, and there's a nice little picture here too. Now we're going to start assigning cells to those kinds of immunity. So T cells respond to intracellular pathogens, which are inaccessible to antibodies and kill cells that harbor them. So these can be very small bacteria. So there are plenty of bacteria in the bacterial biosphere who are capable of entering our own cells. We think of that being a more viral domain of infection, but bacteria do it as well. Viruses are obligate cellular parasites because they don't have the cellular machinery in place to replicate themselves. They need to hijack ours in which to do it. So that's T-cell mediated immunity. For example, if a cell has a virus inside of it, it's going to start pumping out interferons. And an interferon is a chemical that basically signals to neighboring cells that the cell producing it has a virus and they should upregulate antiviral processes if they are its neighbor. Humoral immunity is where B cells produce antibodies. And those antibodies are going to assault and stick to and target for destruction any extracellular pathogens. So this generally disables pathogens by creating clumps of them which are unable to travel, and then those clumps are typically destroyed by the complement system and or eaten up by macrophages. So again, humoral refers to body fluids. So now we're going to get into why these cells have the names T and B respectively. So T cells, which are responsible for, again, cellular immunity, these are born in the red bone marrow, but they mature in the thymus. So the thymus is an organ that is very large in children and increasingly small in adults. And that's because you can think of the thymus as being kind of like the most brutal and intense police academy ever. So basically what the thymus does is it takes immature T cells and shows them self cells and non-self cells. Any T cell that reacts to a self-antigen, so one of the antigens on your own cell surface, any T cell that reacts negatively to that sign is killed. T cells that demonstrate they can tell the difference and preferentially attack non-self antigens are permitted to graduate and move from the thymus into the spleen, tonsils, lymph nodes, bone marrow, etc. So, because the cells of the thymus either graduate, quote unquote, or die, the thymus gets smaller and smaller the older one gets. So, the reason for this maturation is partially due to hormone secretions by reticuloendothelial cells in the thymus, which basically stimulate the T cells to produce surface antigen receptors. So these particular hormones have names like thymosin, for example. So with regard to T cell immunity, T sub C means cytotoxic T cells. So these are going to directly attack other cells. And T sub H, this stands for helper T cells. So some of these, each, helper and cytotoxic, become memory cells. So memory cells don't actually participate in any immune fight going on. So let's put non-participants. But rather, they stick around so that they can remember the pathogen for next time. So that allows them to respond to exposure to a pathogen much more rapidly than naive T cells. And upon re-exposure, they launch an attack that's so rapid, so low latency to respond, that even if a pathogen is present in the body, the person suffering from it doesn't have any noticeable illness. The immune system takes care of it prior to them actually feeling sick or getting sick. So medical application, the retrovirus, human immunodeficiency virus, HIV, basically produces an acquired immunodeficiency syndrome, AIDS, and what this does is knocks out helper T cells, specifically a really versatile and important kind of helper T cell, the CD4 helper T cell. So this effectively kicks the legs out from under the immune system, and indeed most people suffering from acquired immunodeficiency syndrome or AIDS don't actually end up dying of the HIV virus. They end up dying of subsequent infections, often nosocomial ones. Nosocomial infections are those which are acquired in the hospital. So things like pneumonia, Pseudomonas aeruginosa, Staphylococcus aureus, stuff that would not be hard for a normal person to survive is very difficult for someone with a disabled immune system to survive. Okay, so I mentioned that rapid response phenomenon. So this is actually showing you the humoral response instead of the cytotoxic or natural killer T cell response. So these are going to be levels of antibody in response to a first and a second exposure. So remember, antibodies, and I'm going to abbreviate them alpha B, antibodies are not alive. They are produced by living cells, these memory B cells, but they themselves are not living. So they're not alive, only proteins, and these proteins float in the plasma. So, in the primary immune response, antibody production takes a while to ramp up because first the cells have to notice there's a problem, and then antigen presentation has to take place, and some changes have to go on in the lymph node. It's a long process. The secondary response, by contrast, now there are memory cells kicking around, and they remember what that initial pathogen looked like, which means they can respond much more quickly. So specifically, immunoglobulin Gs In the initial or primary response, it takes over 5 days, so more like 6 for them to begin to ramp up following the initial exposure event at day 0. Over here, with the very same antigen following that initial exposure, we can see IgGs experience this exponential increase before plateauing off. So this right here, this little exponential curve upwards, is why you don't get sick on second exposure. So those antibodies are going to go in and disable that pathogen before it has a chance to damage you at all, almost. Okay, so the lymphatic system. Now, when I was lecturing in Zoom, I left you guys with a question about this picture. I asked you to note that the right lymphatic duct drains the right arm, the right upper thorax, and the right head. Everything else is drained by the thoracic duct. So what I did was ask you to think about what you know about the pressures generated by either side of the heart, and then come back and tell me, for example, why you think that particular asymmetry exists. So a lot of you thought about this question very hard, and when we returned to the Zoom session, several of you pointed out that there is asymmetry in the pressures generated by either side of the heart. So the right side of the heart, which receives blood from the systemic circuit and sends it next door to the lungs, is a relatively low pressure system compared with the left heart. And so the pressures in those two circuits are different, and you guys guessed correctly, in fact, that that kind of accounts for the asymmetry in the development of the lymphatic system. So you are correct there. And I was actually very, very proud of you guys for drawing that conclusion, because I've asked that question many, many times in the past, and you know, it's kind of hit or miss. A lot of groups of students don't pick up on that, so nice job. So as I mentioned, lymph is an extracellular fluid. It's similar to plasma, but it has much less protein. So for example, most albumins, fibrinogen, are absent from lymph. I don't know why I put an apostrophe there. Whoops. So as we established, that lymph is picked up by lymphatic vessels that intermingle with the capillary beds, and that's the first site where lymph is gathered. And then we have lymphatic tissues and organs. So the distinction here we will get to, but basically the idea is if it's just a clump of lymphocytes, it's a lymphatic tissue because it doesn't really have defined boundaries. If it's a lymphatic organ, that means it's got its own connective tissue capsule and is distinct from neighboring structures and organs and tissues. So that's really the where the line is drawn there. So diffuse lymphatic tissue is the simplest form, and this particular drawing divides it into BALT, which is bronchial associated lymphatic tissue, and then GALT, which is gut-associated lymphatic tissue. And this one would be abbreviated GATALT and NALT. These terms are all very silly. All of these are places that contain mucous membranes, so it's appropriate to call all of them mucosa-associated lymphatic tissue, just as a group. Lymphatic nodules, also called follicles, are dense congregates of lymphocytes, and we'll start to see these a lot in lab when we get to the nitty-gritty of the GI tract, and also Peyer's patches, which are lymph node clusters in the small intestine. So as I mentioned previously, lymphatic organs have their own connective tissue capsules. So dense, irregular connective tissue, as is typical of connective tissue capsules, and this helps to isolate them from neighboring tissues. So of the organs we've got primary and secondary. So the difference in these is just the site where they learn to be immunocompetent. So remember, being immunocompetent means that the cells can recognize and distinguish between and among antigens and respond to them appropriately. It also means that they will not attack self cells, so the body's own cells. They won't display autoimmunoreactivity, which is not ideal because, of course, you don't want to be attacking your own cells and tissues. So, primary lymphatic organs are the sites where these cells can become immunocompetent, and those are going to be the red bone marrow and the thymus. So remember, both T and B cells are born in the red bone marrow. T cells migrate to and mature in the thymus. B cells just stay in the bone marrow until they navigate to the spleen. Secondary lymphatic organs are populated by cells that are already immunocompetent. So cells migrate from here in the primary to there. So lymph nodes, tonsils, and or the spleen. So I discussed the thymus earlier, but this is just a picture showing before and after involution. So it sits on top of the heart, and as you can see, the difference in size and histology between a juvenile thymus and an adult thymus is quite obvious. So a child's thymus has multiple lobules, which are these big sort of projections. And in between the lobules are septa. And septa contain sinuses and also connective tissue. Also, each lobule has a cortex, which is this outer dark blue layer, and a medulla, which is this inner light blue layer. So those tissues color differently because they are kept separate from each other. So immature and not yet immunocompetent T cells occupy the outer part, and then they'll migrate into the medulla if they are allowed to graduate. Also, if you look closely, the medulla is full of numerous blood vessels, and those blood vessels are the route by which the mature T cells will leave the thymus and go migrate elsewhere. Now, as thymic cells are either killed or leave, the thymic tissue becomes smaller and smaller. After involution, there's only small regions of lymphoid tissue, and then most of what you see is adipose. So the cortex, which remember is that darker colored outer layer of each lobule, is packed with lymphoblasts, and these are going to be, of course, lymphocyte precursors. And that really just means that they are not yet immunocompetent, and they're working on becoming. These epithelial reticular cells, also called reticuloendothelial cells, they're going to secrete cytokines like thymosin and thymopoietin, for example. That initiate the maturation of these surrounding T cells. So they're going to act on their neighbors to cause them to mature and grow up. So Hassel's corpuscles, also called thymic corpuscles, are these concretions or aggregates, and they're partially mineralized, and they're partially epithelial cells, and they're partially also vascular tissue. They're this weird hodgepodge, and they occupy the medulla. So they have epithelial cells releasing cytokines, so again, thymosin, thymopoietin, prostaglandins, etc. And they're clumped together just for efficiency purposes. So they're going to radiate hormones out from their centers, which are going to do things like stimulate the nearby cells to mature. Okay, structure of lymph nodes. So lymph nodes in general, they're situated in many places throughout the body, as you may have seen from the image I provided you with earlier in the PowerPoint. So lymph node, I analogized in lecture during Zoom, is kind of like IKEA. And you might be wondering, what the hell are you talking about, Professor Howard? Listen, you had to be there. Just kidding, I'll explain. So if you've ever been to IKEA, and I'm talking about specifically the one in Portland, although I would imagine they're all designed this way, believe it or not, I've only ever been to one IKEA, and it's ours. If you go to the IKEA that we have, there are a couple ways to enter. So you can go through the main doors and up the escalator, or you can go through the doors by the little snack bar downstairs and up the escalator, or you can go in by the return section and up the escalator. Regardless of how you get in, once you do get in and up that escalator, there is no down escalator. So the only way for you to get out of the IKEA is you have to walk by all of the stuff available to buy, which from a marketing and sales strategy standpoint is genius because, I might enter IKEA with the strong conviction that I'm not going to buy anything today, but odds are I'm going to be on my way out in that little plant section and see like a cute little plant in a cute little pot and be like, well, I can't leave empty-handed. I might as well get another plant. IKEA is designed that way on purpose. It's difficult to leave so that you have ample time to percolate through the store and work up the urge to purchase things. It works really well. The structure of a lymph node, as you can see, is very similar. So there's lots of entrances and only one exit, which forces the lymph to sort of slide around in and through all of these little spaces in the lymph node, which gives the immune cells that live in the lymph node a chance to observe that fluid and check it for pathogens. So this design is on purpose. So I mentioned the cortex and the medulla previously, but it doesn't hurt to repeat it. So the cortex is going to be the outer region, and this is true for any organ. So cortex just means outer layer. So this is primarily aggregates of lymphocytes, both T and B cells, for example. And then at the sort of germinal center, this guy in the middle of each cortical area is the area where the lymphocytes are actually actively activated. So they may hang out in the medulla, but they're only being activated in the germinal center. So the medulla is in the center here. So I want to stress that this area is the medulla. This is the cortex, which means that the germinal center is a feature of the cortex. So the medulla is the lymphatic tissue separated by medullary sinuses, which are these little guys. So just spaces between cells, that's what sinus means. It's a place through which lymph passes. And then the capsule is the dense connective tissue that covers the whole thing. So the order of operations for flow is afferent lymphatic vessels to the subcapsular sinus, which is just a little empty space underneath the sinus. And then it trickles through the trabecular sinuses and medullary sinuses. So it trickles down from the edge through here. So a trabecula is one of those little septa that poke down between lobules, and the trabecular sinus is on either side of those. So here we can see some T and B lymphocytes, and notice that this is a picture of some brown tissue. Is it really brown? No, but immunochemistry has been done to this to stain for very specific tissue markers that mean specific things. So in A, this top one, the cells have been stained for a T lymphocyte marker. So an antibody, this is what anti means, antibody against. CD3, which is a protein, it's a surface marker. So anti-CD3 is going to show us where in that lymph node the T cells are. So the T cells are mainly situated around germinal centers and in the deep cortex. So these dark brown areas are where they hang out, and around here. Anti-CD20, which is a B lymphocyte marker stain, this results in a picture that looks like this, where the germinal centers themselves are chock full of B cells, but not T cells. So there's actually a spatial distribution and distinction between where T and B lymphocytes are inside of a lymph node. So here we have the H&E stain of a lymph node. So we can see that the subcapsular sinus is this little open space between the capsule itself and the tissue. Now, it doesn't mean it's completely empty. It just has a lower relative density of cells than other places. The trabecular sinuses are the little sinuses on either side of a trabecula. And then the superficial cortex is the one that contains lymphatic nodules. So this includes an area that is around a germinal center. Sorry about that. I had to step away to sneeze so I didn't scream sneeze into the microphone. Yes, sometimes I scream sneeze, especially during allergy season. I bet some of you can relate to that. Anyway, I'm back now, sneeze-free, thanks to Zyrtec, not an affiliate of this show. So where we left off was the superficial cortex. So that's the lymphatic nodule, which I've circled. And then the deep cortex doesn't have any nodules. It's just kind of a dense lymphocyte aggregate zone. So that's most easily outlined by doing, come on, pen, this. There we go. So like that. And in fact, it kind of wraps around here, really. So the medulla is populated by medullary cords, and I know that the lines actually don't do a good job showing you what those cords specifically are, so I'll outline them for you. So you can kind of make them out a little bit better if I give them an outline, like so. And then the medullary sinuses are the spaces between them. So the cords are just columns of cells, and the sinuses are the lighter appearing spaces between them. So basically lymph is going to sort of filter through these things on its way, and that means that the lymph has to bump into and pass a bunch of different cells, lymphocytes primarily, but also macrophages, before it gets out. So giving it ample opportunity to be discovered by the immune system if it contains pathogens. Okay, so when we were talking about tissue types previously, I sort of mentioned that, hey, some stains are better than others for resolving certain kinds of fibers and kinds of cells, and we did talk about reticular connective tissue. And I mentioned that, you know, some stains make the reticular fibers show up extremely well, others less so. So silver stain is one that shows reticular fibers especially well. So a friendly reminder that reticular fibers are collagen plus glycoproteins. So the glycoproteins are significant because this gives the cells that live inside of the lymph node or whatever other reticular connective tissue based organ there is, it gives them something to stick to. So that's kind of the idea there. So it forms an irregular anastomosing network. What does that mean? Well, it means there's no particular order, and the fibers split and then rediverge without any particular purpose or order. So that's what that means. Stroma is just a generalized word for the parenchymal cell population of an organ. So you can see the nuclei over here. And these reticular cells, which are the arrowheads, so they have a characteristic oval-shaped appearance compared with the lymphocytes, who have round nuclei. So those reticular cells, also called reticuloendothelial cells, are going to be in contact with the reticular fibers. Okay, so here's lymphoma. So lymphoma is a type of cancer, and it's specifically a cancer of the lymphocytes, hence the name. So these are going to be logically, or excuse me, not logically, blah, I was looking at the word logically on my whiteboard when I said that. These are going to be sort of overpopulated lymph nodes, so you would get a swelling. You also get these changes in cell morphology. So generally speaking, if you're a pathologist, what you're looking for is not only changes in cell number that are associated with cancer or neoplasia, but also typically the neoplastic cells don't quite look right, and you're seeing that here as well. So these guys you can see have large kind of knobby nuclei with more than one nucleolus, which is unusual. So typically, like this nucleus right here looks a little bit more normal. You see one nucleus and one nucleolus per nucleus. Not so with the cells, the nuclei of the cells I have circled. So these change the normal architecture such that there is no medulla anymore, no trabecular sinuses, no medullary cords, no medullary sinuses either. You basically get a lymph node that is all nodule. So cortical tissue replaces the medullary tissue, and that's not normal. Unfortunately, lymphoma has a high likelihood of metastasis because lymph nodes have such good access to the lymphatic system and blood supply, meaning that cancerous neoplastic lymphocytes can hop into the bloodstream relatively easily and travel elsewhere in the body. Not ideal. So you tend to have lymph nodes or lymphoid nodules that are guarding entrances and exits to things. So one example is the pharynx. So here we have the nasopharynx and the oropharynx. And then from the tip of the epiglottis down to the start of the esophagus, we have the laryngopharynx. So the reason that you have pharyngeal lymph nodes is because there's a lot of stuff that passes through this area of your head. So for example, you breathe through both your mouth and your nose depending on what your activity level is. So pathogens can travel in the air, we know that. And of course, you also eat and drink fluids through your mouth, so it's another way for pathogens to get in. So from that perspective, it makes a lot of sense to have guard stations at those openings. So the palatine tonsils, meaning at the palate, are posterior. And you have, generally speaking, one of these on each side. So you'll say ah and look at your uvula, and then there's tonsils on either side. The lingual tonsils are at the back of the tongue on the root, so down here. And then the pharyngeal tonsils are situated on the back of the nasopharynx, not far from the opening to the eustachian tube. And because the pharyngeal tonsil is in the nasopharynx versus the palatine and lingual, which are in the oropharynx, the pharyngeal tonsil tends to have epithelium that matches that of the nasopharynx, so ciliated pseudostratified columnar, whereas the tonsils in the palatine and lingual regions are covered with stratified squamous epithelium because that is what dominates the oral cavity. So, same general function, different epithelial covering according to location. So if you get chronic inflammation of the tonsils, which many people do, you end up with pharyngeal hypertrophy. So the tissue of the pharyngeal tonsil becomes large due to chronic inflammation and cytoarchitectural changes because of it. And the specific name for that pathology is called adenoids. So here is one reason why people get chronic inflammation. Tonsillar crypts are these invaginations of the tonsil, and these fill up with all kinds of gunk. So epithelial cells that are no longer squamous, live-ended lymphocytes, bacteria. So there's little wars being waged in your tonsils, in your tonsillar crypt specifically at all times, as well as just cell buildup. Someone during the Zoom lecture asked about the idea of tonsil stones, and I was just like, hey, sometimes cells build up faster than normal in these crypts, and then you can pop them out, and that's what a tonsil stone is. Pretty gross, but it's a thing that exists. So as far as architecture goes, it's pretty similar to a regular lymph node. We've got nodules with germinal centers and then some medullary tissue, which you can't see on here. But the nodules are especially numerous along those infoldings and along the crypt. So this allows close monitoring of the contents of the crypt by the immune cells populating the nodule. So, The epithelium surrounding the tonsillar crypt, accordingly, can become difficult to recognize because the surrounding lymphocytes kind of migrate inward, and that's what you're seeing here. So you see these little cells with the clear cytoplasm around them sort of making their way through the epithelium and ending up embedded within it. So as you can see, I'm drawing an outline around the boundary that demarcates the actual lymphatic tissue from the epithelium. So the epithelium is from here to here, so I'm making a little bracket, and then underneath this is the actual lymphatic tissue. So you can see that there's a lot more clear space here, so dark little nuclei with clear cytoplasm around them, and you can easily see how this cell would migrate into here and look a lot like that cell. So chronic inflammation of the pharyngeal tonsil specifically produces adenoids, and because those particular tonsils are right by the auditory tube, which connects the nasopharynx to the middle ear, it can end up causing what is called otitis media, which is middle ear infections. And that is characterized by the tissues and structures of the middle ear, so it's an air-containing chamber bounded by the tympanic membrane, becoming infected and weepy, and sometimes filling up with pus, which is very gross. So the spleen is an interesting organ because it's kind of a double-edged sword in a weird way. It has some very peculiar features, and I will explain. So if you get the procedure written down here called a splenectomy under safe medical supervision, you can live just fine without a spleen. It's not a life or death organ. However, if your spleen gets squished or stabbed or lacerated, anything that ruptures the capsule and ruptures the tissue, that's going to require immediate splenectomy because you will bleed out internally if it is left to its own devices. And this is because the spleen is extremely vascular, so its blood supply is gigantic, and that's partially because, of course, it has to check the blood and the lymph for things. It's also a place where blood is stored, where red blood cells go to die, and where blood is produced in certain fetal developmental stages. So your spleen's a really big deal. It's very, very fragile, but if you happen to get it removed, other organs can take over. So the white pulp in the middle, which is this not red stuff right here, is primarily lymphocytes and macrophages, and what these are going to do is surround branches of this splenic artery and monitor the blood for foreign antigens. So basically, it's kind of like a racetrack. So if you think about a racetrack is a place where people drive, and then on the sidelines there are folks watching the drivers. Same idea with the white pulp. So they're going to cluster around these speedways that are the blood vessels and watch what passes by to make sure everything is okay. So here's the general structure of the spleen. As I mentioned, the white pulp is lymphocytes, and let's not forget macrophages. And expansion of this creates the splenic nodules. So this looks like lymphatic nodules, but they're deep in where the medulla would be on a lymph node. The red pulp basically sort of occupies the spaces between the white pulp. So you can see it's reddish in color because it's very rich with blood. And it's surrounded by splenic cords, also called the cords of Billroth. So basically just tissue surrounding these pulpy areas. This is a very blood-rich area. So blood vessels traverse the capsule and the trabeculae both before and after passage within a spleen. So you can see that they enter, branch, and then branch some more, and then leave. So again, what this serves to do is to provide an opportunity for any interstitial fluid containing pathogens to be exposed to lots and lots of immune cells, with the understanding that if they are bad and remembered, they will be recognized. Doesn't always happen, but it happens that way fairly often. All right, so blood circulation and the structure of the spleen. So why is it so sensitive to trauma and rupture, and why will it make you bleed out in a jiffy if your spleen gets hurt? Well, The way that the blood vessels are arranged facilitates monitoring, but also makes the spleen vulnerable to trauma. So the way this works is, and you can see that there's arterioles, or arteries and arterioles on the right, and veins and venules on the left, just in this zoomed in area. The central arterioles are enclosed by lymphoid cells. So basically, remember I said it's like a racetrack where the blood cells, or blood vessels, excuse me, are the racetrack and the cells surrounding are the spectators. This is where that happens. So the spectators, so to speak, have a name, and that is PALS, which is cute, right? This really stands for periarteriolar, fun word to say. Say that 5 times fast, I dare you. Lymphoid sheath, or PALS. So think of these as a bunch of cells that are looking into the central arteriole and watching what is there. So B cells in these sheaths can form nodules, which is what this white pulp nodule right here is trying to show you. So blood either flows into a closed circulation or an open circulation, which is where it either goes into splenic sinuses or it's just dumped into the lymphoid tissue. So the blood sort of sloshes around in between cells in the red pulp's splenic cords. From there, the cells re-enter the vasculature through walls in the sinus. So this is a very unconventional blood circulation plan. So open circulation is limited to very, very few areas of our body. In general, in mammals and vertebrates, our blood should be in our blood tubes. If it's not, that's a problem. The notable exception here is, of course, the spleen. So the only place your blood should be kind of sloshing around between the cells, not in a tube, kind of not doing anything tube-like, is in your spleen. Anywhere else, that's bad. Oh, let me just erase this. So here is a follicular lymphoma of the spleen. So normally spleens are supposed to be this sort of dark livery red color here. These numerous white patches, and this is a fresh section, by the way, so not stained in any way and not prepared histologically, just as is. It is not supposed to look like this. So all of those little white pulp follicles that are expanded are basically indicators of pathology. Now, you wouldn't be able to tell whether this was a particular kind of lymphoma. So if you look at the caption here, it says other indolent B-cell lymphomas, mantle cells, marginal zone, etc. So there's multiple kinds of cells that can basically go rogue, so have some kind of dysregulation of the cell cycle and produce this particular pattern. So in order to find that out, one would need to use immunocytochemistry to stain specifically for the cell type. And that is what a pathologist would do in this situation. So this is just a nice summary slide showing you the different lymphatic system levels of organization. So everything from diffuse lymphatic tissue to full-blown organs like the spleen. So what I would do here is not use this as your be-all and end-all, but rather use this chart as a way to sort of organize your thoughts as you're studying for and taking the exam. Okay, so the digestive tract. We're moving on to chapter 15. And if you need a crash course in any of that, the links at the top left are useful to you, but I, of course, am not going to click them here. So with regard to anatomy, the digestive system has 2 anatomical subdivisions, and these are the digestive tract, also called the alimentary canal, because aliment is food. And this is actually a French borrowing. So in language, in English, we have Latinate borrowings, we have French borrowings, and we have German borrowings. So our language is kind of a mashup of a bunch of different languages. That's why it's so hard to learn. So this is one of the places where we were like, we like this French term for food, so let's apply it here. So the digestive tract is the 30-foot long muscular tube extending from mouth to anus. So although humans think of ourselves as these fancy, super well-planned, thought-out, complex organisms, we're really a just kind of stretched out torus. So a torus, if you're not familiar, is the geometrical term for this shape, which is a donut. So there is a, let's make a contrasting color here, empty space in the middle, and then tissue on the sides. It's just that if you stretch the torus out long enough and you kind of wrinkle up the inner tube space, you get this. Interesting fact, humans are deuterostomes, which means mouth second. So stoma is mouth, deutero means second, or the second one, which is a nice way of saying anus first. So all humans, let's make an asterisk here, form ass first. And yes, I said ass. It's my YouTube channel. I will say what I want. So protostomes form mouth first. We do not. Came into this life butt first. Probably will leave it that way as well. Go deuterostomes. The gastrointestinal tract is limited to the stomach and intestine, so it doesn't include the esophagus or the mouth. The reason for the distinction is that different phases of digestion and absorption happen in different areas of this particular tract, so that's why. Accessory organs include teeth for mechanical digestion of food, tongue also for that, as well as for forming speech, salivary glands for saliva, the liver, the gallbladder, and the pancreas. So most of the accessory organs exist to change food, to prepare for absorption. So the 5 stages of ingestion are as follows. Digestion. We've got ingestion, which is the selective intake of food. So that's just a fancy way of saying, like, you don't eat anything. You eat things that are A, possible to eat without dying, and B, which provide you with nutrients in order to stay alive. Digestion is taking the food that you choose to ingest and shredding and grinding it up, and also chopping up molecules using enzymes so that the food is in small enough units that you can use it. So for example, I can't take a cow muscle protein and just replace one of my own muscle fibers with it. I would need to grind it up and then use enzymes to break up the polypeptides, and finally absorb the amino acids that result so I can reassemble those amino acids into my own muscle fibers. That's the key there. Absorption is the uptake of those nutrient molecules. So I just mentioned amino acids. Basically, they have to cross into the epithelials, and then from those epithelial cells into the blood or the lymph. So here's things that go into the blood. Amino acids, carbohydrates, so these are saccharides, monosaccharides and disaccharides primarily, so let's write that. So basically anything water soluble. Excuse me, dainty cough. Lymph is for fats. So there's a little bit of a difference as far as how fats are dealt with from an absorption perspective than there is with amino acids. This is also including nucleic acids and monosaccharides. Those are all things that are polar and therefore can just be absorbed by themselves into the blood because they are water soluble. Compaction is basically reclaiming water from what you've eaten and the water that you yourself contribute to your own digestive lumen. So if we go back a slide, which I'm going to do really briefly, you'll actually see if you look carefully that you're secreting water into your own gastric and intestinal lumen. So 1,500 mils here, 15 here. So this is 3 liters of secretions Now we're at 3,500 milliliters, so 3.5 liters, and then add 2 more, means 5.5 liters of your own body water you are putting into your GI tract and then taking back out during compaction. So this is why staying hydrated is important, among other things. You need that water to be able to digest your food. And then, of course, whatever's left over after you use up all the nutrients and take back all the water, that is what is eliminated as feces. So these are little nice summary cards that show you the differences between carbohydrate digestion, protein digestion, and other particular macromolecules, basically. So what we're doing when we eat is we're consuming useful polymers that we break down into monomers or dimers, and then use those monomers or dimers to rebuild our own polymers. So I'm not going to read these to you because you can read them yourself, but you are responsible for knowing, for example, that salivary amylase is going to turn starch into oligosaccharides and disaccharides. So be able to associate ACEs, enzymes of various kinds, with their locations, so salivary glands, pancreas, and then know the step-by-step dismantling of each polymer. Of course, if you have questions about that, feel free to email me or get at me on Slack. Either one is fine. I'm just telling you that it's not a good use of my time to read you this because you can read it yourself. But anything that you're confused about, I would be happy to answer in lecture or via email. Either is fine. So I mentioned fat digestion is a little bit different, and that's because fat is not water soluble. So you need this extra step, which is emulsification. So if you're confused about emulsification, think about the following. If I have a bottle of vinaigrette salad dressing that's been sitting in my pantry unopened, and I want to use it, I'm going to open it, and then what am I going to do? Shake the living hell out of it, right? Why? Well, I'm going to shake it so that the oil becomes tiny little micelles that are suspended in the aqueous vinegar portion, and that's the only way it tastes good on my salad, if it's mixed together. So that's emulsification. It won't stay that way because I don't have any chemical emulsifiers to do so, but bile salts and the detergent action of them do have that power. Okay, so the basic structural plan of the digestive tract wall. So this is a way to talk about the various layers of tubes of muscle that basically holds true for the rest of the course. So we're starting talking about this in the digestive system, but it's also true for the intestines, and it's also true for the urinary and reproductive tracts, as well as for the vasculature. So mucosa, submucosa, etc. These are basically layer names. So mucosa is the innermost layer, so we're going from center of the lumen outward. And this consists of epithelium, and then the underlying lamina propria, which is loose connective tissue. Sometimes that has malt. The muscularis mucosae, which is a thin ring of smooth muscle that surrounds the lumen. The submucosa is under that, and this is primarily dense irregular connective tissue and loose connective tissue containing blood vessels, lymphatic vessels, nerves, and glands of various kinds. In fact, submucosal glands are often a good thing to look at in lab when you're trying to, for example, decide whether you are in the duodenum or the ileum. Hint, hint, duodenum has Brunner's glands, ileum does not. And then after that, there is the muscularis externa, which has an inner circular layer, and in certain places that thickens up to form valves, and also an outer longitudinal layer, which is going to run parallel with the direction of the tubing. So this is going to propel food through the tract. Together, these two allow for an action called peristalsis. The very, very outermost portion of this is called serosa. Now, most of the organs in the abdominal pelvic cavity are surrounded by serosa, which folds back on itself to form the mesentery, which is a loose connective tissue and squamous tissue sheet that wraps around all of the organs and also contains things like lymph nodes, fat, lymph vessels, veins, and arteries. So it's also an anchoring substance. So the nerve plexuses mentioned here, these are similar to, so here's the myenteric plexus, for example. The nerve plexuses are similar to the conducting system of the heart, in which they have automaticity. So they are going to generate action potentials spontaneously, so by themselves. They are modulated by the autonomic nervous system, but not told what to do by it. They generate their own specific plan of action, and they can operate independently of the central nervous system as well. So this is going to promote gastrointestinal tract motility, meaning that the rate of peristalsis goes up, or reduce it depending on what's going on with you. So this is actually an explanation for why people's digestion rate changes under, for example, stress. Okay, so the oral or buccal cavity. Yes, it's spelled B-U-C-C-A-L, and it is pronounced buccal, not buccal. So the buccal cavity basically is for a bunch of things, including food intake, but also grinding up food, responding sensorially to food, so taste, as well as using your tongue to swallow, your tongue, lips, cheeks, and palate to form speech. And if your nose isn't cutting it, you can use your mouth for respiration as well. So there is a stratified squamous epithelium lining to it. And in areas that are subject to food abrasion, so think of a rough food like kale, for example, you've got keratinized stratified squamous epithelium, and that is because the cornified layer at the top is hardier. In other layers, you've got non-keratinized. So these are the softer parts of the mouth, floor of the mouth, the soft palate, the insides of your cheeks. So this is a place where you can, for example, take cheek cells from if you swab vigorously. So with regard to medical application, viral infections cause the death of infected epithelial cells, and that leads to blisters. So we call these vesicular ulcerous lesions. And I do want to stress that although simplex 1 is usually associated with oral lesions and simplex 2 with genital lesions, you can get herpes simplex 1 on your genitals, and you can get herpes simplex 2 in your mouth and on your throat. So herpes does not discriminate. All it wants is for a cell to infect and then a neuron to hide in. It doesn't really care where on your body that occurs. Also, most humans have some form of herpes simplex, whether they're symptomatic or not. And so I think it's important to normalize the idea that, because herpes viruses are among the most successful group of viruses in history, and they don't discriminate, and they tend not to kill their hosts because that's bad parasitism, herpes is really, really common. So it's talked about by a lot of people as being this sort of thing that people have who deserve to be socially ostracized. That simply isn't true. If that was the case, everybody can just go hide in a hole because almost everybody has some form of herpes. The problem with herpes is that it's opportunistic. So after an outbreak, it will retreat back into the neuron from whence it came, and it will go incorporate its viral DNA into the DNA of the host cell. And there it stays dormant and just waits. And what it waits for is any time when the immune defenses are weakened. So this is a little guy that hides in the shadows and only pops up his ugly head when your immune system is crashing. So here we have the tongue. So the tongue is covered with papillae. Papilla means nipple. Yes, you have tongue nipples. Just kidding. In anatomy terminology, anything that even looks vaguely nipply is called the thus and such papilla, which means thus and such nipple. And that is a naming convention that exists from the Ars Terminologica and Anatomica Terminologica as well, which is an international governing body that all decide on the right way to call anatomical thingies. So filiform, foliate, fungiform, and vallate are names for different shapes of papillae. So the filiform are the dominant kind. These are on the surface of the bulk of your tongue. And these perform frictive functions as well as helping with taste. Foliate papilla are on the sides of the tongue, and these are extremely well developed in young children. After a lifetime of rubbing on your teeth, they're not as sensitive in adults. Vallate papilla are V-shaped, or they're arranged in a V-shaped line, rather a chevron, so to speak, near the back of the tongue. So taste buds are present on fungiform and foliate papilla, but they're much more abundant on vallate. So that means that you really taste the best at the back of your tongue. Gustatory cells themselves are arranged into these little buds, which are shaped like an onion. And they have little hairs with lots and lots of receptors on them poking out into what's called a taste pore. So the taste pore takes dissolved saliva with stuff in it, stuff that you eat, and it exposes that stuff to those sensors on the taste hairs. If sufficient amount of a tastant binds to receptor, you get a change in membrane potential and excitation of that particular neuron. So that's how tasting works. The esophagus is a thick stratified squamous epithelium, and this is because the esophagus is subject to a lot of abuse. So not only do you vomit through your esophagus, which hurts and is bad for it, but also we don't eat food that's always perfectly smooth. So we need to be able to spare some cells if we're going to keep a healthy esophagus later into life. So it's got a thin lamina propria underneath of that, containing occasional lymphatic nodules. I can show you a couple. So here's one, just to name a couple. Here's another one. And then the muscularis mucosae, which is on the outside. So you can't see it here. This particular picture is too far zoomed out. The submucosa contains mucus glands, and that makes sense, right? Because think about a time you swallowed something that was like too dry, how difficult that was. Oh, excuse me. You don't want that to be the case. You want to have a smooth swallow with no stuff sticking in your throat at all times, as much as possible. So the submucosal glands basically just lubricate the muscular passage of the esophagus. The muscularis externa, which I'm bracketing here, are the two layers of smooth muscle again. So inner circular layer, outer longitudinal layer. The esophagus is behind the trachea, and it's between your trachea, so your windpipe, and your vertebral column in your thorax, meaning it doesn't have a lot of wiggle room. So it is stuck to the vertebral column by adventitia, which helps to anchor it in place and keep it from rolling around as it tries to wrestle with the large bites of food we all take. So here's another picture of the esophagus. You can see a collapsed stellate lumen, meaning star-shaped. So the esophagus is also quite stretchy to change in size to accommodate many, many different sizes of food. Anybody ever been in the position where you swallowed something that was too big and you could feel it going all the way down? That's what that feeling was. It was this esophageal lumen stretching, and probably you're losing some cells from here. So the esophagus basically just transports swallowed material from your larynx to your stomach, avoiding, of course, the airway. So this is a picture showing what happens when the lower esophageal sphincter fails. So ordinarily it's supposed to keep acidic stomach contents from sloshing back up and damaging the epithelium of your esophagus. But if you have scar tissue due to acid reflux and heartburn in a prolonged or chronic fashion, you can get a stenosis, which means a persistent opening of the lower esophageal sphincter, and that is full-blown GERD, or gastroesophageal reflux disease. So this often requires surgery to address, where they go in and they basically tighten up this little sphincter for you so that your reflux doesn't persist. Having repeated exposure to acid in the esophagus is not ideal because it can cause metaplasia and eventual cancer of the esophagus, or an entire casting off of the esophageal mucosa, which is a condition called Barrett's esophagus. So when your esophageal epithelium turns cuboidal and then begins to slough, that's Barrett's esophagus, precursor to esophageal cancer. Okay, and this is the slide that we stopped at in lecture, so this is where I will stop here. I don't feel like I need to really go into this picture. This is just showing you the difference in epithelial types between the stomach lining and the esophagus, and you can see that the transition is really abrupt. It's right here. So we go from stratified squamous to simple columnar with no smooth transition zone. It's pretty binary as far as a switch goes. So with that, I will thank you for your attention and get this to uploading so you guys can have it. I appreciate your time and attention, and I will see you in the next video.